I'm Ranamay Saha. I'm from IIT Kanpur, India. So today I'll be presenting on impedance spectroscopy-based detection of viral RNA from clinical samples. Now, having said that, by viral RNA, it's a generalized term, but initially our point of application will be on hepatitis C viral RNA. And so I will just introduce to you the state of HCV infection across the globe. Around 3% of the world population is currently infected with HCV, and out of that, around 80% have actually chronic HCV with them. Now, out of that, around 20% is actually suffering from cirrhosis of the liver. But the paradox is merely they know about their infection. Around 19% of the people carrying chronic HCV are actually aware that they are having that HCV virus with them. And around 30% of them are actually having treatment. So it actually emphasizes the matter of concern here. But the good news is that WHO as well as national program guidelines has decided to eliminate HCV from the world by 2030. Now, so what's the need of the R? It's to start early mass detection. So that way a rapid detection scheme needs to be developed that can actually offer acceptable sensitivity as well as accuracy, still maintaining the simplicity of the mechanism and also a low cost associated with it. So just having a short look at the current detection schemes which are available, we have antibody screening technique, which is basically based on antigen antibody chemistry. But the issue is that although that offers quite quick results, but they can offer very large number of false positive and false negative results. Also, liver function test can be there, but that makes no sense because already the infection has reached the level 2. And then the gold standard for the industry is qPCR tests, quantitative PCR techniques, all of you must be aware of. But although these are confirmative, but the issue is they involve expensive in-process requirements and also sometimes take up a couple of weeks to actually give the results. So based on that, we have decided to develop an electrochemical biosensing mechanism. If we look at the literature, they offer a decent level of accuracy, but still they can give quite quick results. Sometimes within minutes or an hour, they can give out the result. So having said that, we have decided to develop something based on electrochemical impedance spectroscopy. So it would be a rapid test. It should be able to give accurate results while maintaining the sensitivity. At the same time, the cost associated should be low. And then early detection is of course the prior thing for any rapid tests. So if we look at the literature quickly, we can see that researchers have been working on it since the last decade maybe. They have been able to reach up to femtomolar range of LODs. But the issue is that most of them have adopted faradaic electrochemical biosensing. So the faradaic sensors have the issue that they need some biomarkers for the detection. So that is a complex thing, and often it becomes difficult to arrange for that complementary biomarker that can help us in detecting. So second issue is they have used around 10 microliters of analyte volume sometimes. That is not very much low in the context of micro nano engineering. And the third issue is that the integration level of these sensing schemes are quite low in most of the researchers. And sometimes they don't even mention what is the integration level. So having said that, there are three major issues which I have mentioned, and we have framed our objectives based on those issues. So our chip should be a compact one so that we can make it portable. We can carry it to any of the local regions or any of the suburbs we want to. In that case, we should also be able to get a sufficient level of sensitivity out of the chip. So signal amplification is a factor. So we have taken two amplification steps. One is gold nanoparticle labeling, and the second one is dielectrophoretic concentration of the analyte. So Using that, we have to frame the first objective like standardizing the nanoparticle preparation and then standardizing the nanoparticle DNA conjugation protocol. Second objective is RT-PCR and PCR experiments, data collection and optimization. And the third one is, of course, design and fabrication of the chip, while looking out how we can integrate the concentration step as well as the nanoparticle labeling with the impedance measurements. So we have this the HCV virus DNA, which actually is transcripted from the RNA and is supplied to us from Institute of Liver and Biliary Sciences, ILBS, New Delhi, India. And this shows the primer sequence. I've shown it in the slide. And we have used standard protocols for nanoparticle synthesis, Turkiewicz method. It's a very commonly known nanoparticle synthesis scheme. And we have used Staten's protocol for DNA nanoparticle conjugation. So I'll not go into details because these are standard protocols. So this I'll directly come to our chip fabrication method. It is a 4-layer chip. We have 2 interdigitated electrode layer fabricated on silicon. These are separated by a sandwich layer of silicon dioxide. It will act as an insulation layer just to separate the 2 layers, because the 2 layers will be used for 2 different things. The top layer will be a microfluidic channel layer in which we have designed the channel such a way that it can hold only 100 nanoliters of fluid. And then we can also see the electrode design. We have around 1 × 1 cm square of total electrode surface area. We have an electrode arm of 55 microns and an inter-electrode gap of 40 microns. The IDs consist of 30 nm of chromium, which acts as an adhesive layer, and 120 nm of nickel electrode layer, which acts as a conductive layer actually. So our experimental setup is shown in this slide. We have a thermocycler. It's an append of thermocycler where we do the PCR experiments. We do 20, 30, and 40 cycle of PCRs as of now. And then we take out the analyte out of the thermocycle, and then we just flow it over the top layer electrode. The top layer electrode is connected to the impedance analyzer, through which we take the impedance readings. And then the bottom layer electrode is there, which is actually connected to the waveform generator. You can see the waveform generator that actually does the DAP manipulation of the analyte in the solution. So after we have flown the analyte over the chip, over the top layer electrode, we turn on the waveform generator, we do the DAP concentration, and then we stop, we seize the waveform generator and take the impedance readings out of the impedance analyzer. So here is a visual representation of how the chip looks and what's the actual size of the chip in terms of one of our pump. So you can see that it is quite compact and is very much portable in nature. So then what we actually do the DEP characterization first. For that, we check at what frequency we are getting the maximum double layer capacitance value. The double layer capacitance is actually displayed in the impedance analyzer. So after we have optimized the frequency at which we get the maximum double layer capacitance value, we set that frequency as the DP frequency. And then we take the impedance readings over a wide range of frequencies starting from 20 Hz to 10 MHz. So we then plot in terms of Bode and Nyquist plots, and we can just analyze the events. So looking at the results, first we want to just validate that the conjugation of the nanoparticle with the DNA has taken place. So for that, you can see figure 5B to D. These are transmission electron microscopy images. So figure B shows the scattered gold nanoparticles in the solution. It's just a nanoparticle solution. Figure 5C shows the conjugation when nanoparticles are conjugated to DNA primers. So we can see 1 or 2 discrete clumps in the solution under TEM microscopy. And when we have done the TEM analysis of the PCR products, we can see like a large clump of moiety is there. So that actually happens due to the electrostatic forces between the negatively charged DNA and the gold nanoparticles in the solution. So that actually validates that the conjugation has taken place, and it can also be found in the literature. People have earlier done that. So this slide shows the UV-Vis spectroscopy result. We can see a red shift of 6 nanometers. That actually indicates the increased density, and hence the conjugation gets validated. So we come to the impedance measurements. We can actually easily see here reducing value of the impedance magnitude with increasing number of PCR cycles. That mainly because of increasing number of charge carriers with increasing number of PCR cycles in the solution. And that flow of charge actually reduces the impedance, the charge transfer resistance to be more specific. And further, with gold labeling, it further reduces, the lowest being 40 cycle gold level PCR, in which the maximum number of charge carriers are present. So this is the reason for which that reduction happens. With increasing number of charge carriers, there are increasing number of ion clouds and increasing number of dipole moments, which are present in the solution. And this actually leads to flow of charge, more and more flow of charge with increasing number of charged particles in the solution, and it actually reduces the impedance value. So this is a Nyquist plot. You can more easily visualize what's happening. There are reducing number of arcs. The lowest being the orange arc at the bottom. It stands for 40 cycle gold level PCR. So it's very easy to visualize how the impedance is reducing with the increasing number of particles in the solution. So one thing to note is that the very high frequency region that corresponds to the bottom left corner of the graph, where we can see there is some anomaly, like the impedance is rather increasing with increasing number of PCR cycles. So that can be explained by the fact that because the frequency is very high, the time available for DNA hybridization was very low. So there are some large number of single-stranded DNAs which couldn't hybridize, and because they have very low charge associated with them, they couldn't form the dipoles and couldn't form the ion clouds in the solution. They just accumulated and resisted the charge flow in the solution. So as a result, yeah, sure. So that's the reason. And then we come to the point why DEP was used as a signal amplification step. So DEP actually concentrated the moieties in the highest electric field regions within the interdigitated electrode. And as a result, we could see significant reduction in the impedance values when DEP was utilized as an amplification step. The lowest impedance being the DEP assisted gold level PCR for 20 cycle PCR product. So yeah, that's what we have done. We have successfully detected HCV RNA. Actually, we detected HCV DNA, which was transcripted from RNA. And then as of now, we have gone to 30 nanogram per microliter concentration level, which comes to around 130 nanomolar range. We can further go down with optimization of the process parameters, as well as some additional technology which we can add further add to our current technology. So these are preliminary results which we have got. So this project was funded by Indian Council of Medical Research, ICMR. Yeah.